So I drew a bunch of diagrams prior to this because I'm trying to make it not super long. Um, we're going to start with the pressure and mechanisms that make negative pressure ventilation possible. And there are quite a few parts of the lung that make this possible. So it's kind of complicated, but once the picture is all drawn, it's really not that complicated, which is nice. So our lungs have these tubes called bronchioles, or well, it's bronchi, and then they're bronchioles when they're smaller. So I guess I should call them bronchi. Um, and they have like these nice cartilage rings that keep them from collapsing. So when they're nice and wide, they have these cartilage rings. But then when we get smaller, they're lined with smooth muscle that can constrict or relax. Um, and then we get to these sacs of air at the bottom. This is called the alveolus. When we talk about them in plural, we call them alveoli. And they are where actual gas exchange occurs in the lung. So they're like the functional unit of the lung. That's like the equivalent of the functional unit of the lung, or like the equivalent of the functional unit of the kidney, which is a nephron. This is like the lung's nephron. It does lots of work. The other tubes and the respiratory tree really just gets air to the alveolus. That's the important part, that we need that the rest of the respiratory tree for. It's just making sure that we get air to the alveolus. I'm gonna redraw the lung, and but I'm going to redraw the lung. I mean, I did redraw the lung right here. This black part in the middle is just one big alveolus. Obviously, we can acknowledge that's not what a lung looks like. We have millions of small alveoli, and they work together. Um, but to simplify and make this easier, I drew one alveolus. Then so we have this tube, right, where we come. If this is like our trachea, we have this tube that is going to bring air into our lung. And there are membranes that surround our lung that help us to establish negative pressure. One of them is the pleural membrane, and the pleural membrane is on the inside of like our rib cage. Um, so like we could draw our ribs right here, if these are your ribs, which are terrible drawings of them, but you see the point. So if your ribs move out, like when you inhale, your pleural membrane will also move out, right? Then we also have what we call a visceral membrane right here along the line, along the lung. Oh, now I can't spell because I was talking. Dang it. And this visceral membrane just goes around the lung, and these two membranes work together to create a pressure. So if you imagine like you squeeze your hands together really, really tight, and then you try to bring them apart, and there's like kind of a suction there, you can like almost make a sound with it. That's what happens between the pleural and the visceral membrane. They get really, really tight, and they literally get all the air out to the point where, compared to atmospheric pressure out here, if we call atmospheric pressure zero, which we do when we're talking about the lungs because atmospheric pressure changes. So if we just set it to zero, it makes talking about the lungs a lot easier. This space rests at about -3, right? So it's like suction cup, like it's like a vacuum, like it's like suctioning, it's holding, it keeps the lungs from collapsing because naturally the lungs have this tendency to collapse. They want to fall inward. So with that negative pressure, we keep the lungs just expanded enough that we're not like expanding aggressively, but we're not falling inwards. And this happens by we have like a pleural fluid. Um, that basically like what you could describe it or use to describe it is if you had two sheets of plastic wrap and you put them together and you put like 2 or 3 drops of water in it and then you rubbed them together, right? That would be these two membranes. So really they're not far apart. They're not filled with a lot of fluid, but there is a negative pressure. They're really, really suctiony. So they like kind of suction the lung out. They pull the lung out. And when we inhale, what happens is our rib cage flares outwards a little bit and it moves out, which moves the pleural membrane out. I'm moving my hand like you can see me. You can't. Moves the pleural membrane out right here, right? So all of this moves out, which pulls on that suction, and we actually decrease the pressure in here. So when we inhale, we go from like -3 to like -8 or -10 even. Which then is going to pull on our lung and make our lung a little bit bigger. Our lung follows, oh my gosh, why can't I remember this guy's name? I want to say it's Bohr's law, but Boyle's law. There we go. Okay. Our lung follows Boyle's law. And Boyle's law is that volume is related to pressure in an indirect manner. So as volume increases, pressure decreases. So as we pull on our lung, because we have this decreased pressure in our pleural membrane, we increase the volume of our lungs. So we're pulling the visceral membrane in our lung out as well, right? So our lung gets pulled out a little bit, and the pressure actually in our lung decreases. So we get our pressure down to like -3. And before it was 0, the same as the atmosphere. But when we get our lung pressure down to -3, we're able to inspire air. We can bring air into that lung because pressure follows its concentration gradient, just like a solute would. If there's less pressure on the inside than there is on the outside, we're going to put air in until that pressure evens out, until we have even pressure. Then, when we expire air, we do the opposite thing. So we're pushing our rib cage in, we bring our diaphragm up. I kind of forgot to draw the diaphragm, but we do have a diaphragm. It pushes up on the lungs when we exhale, and it actually goes down when we inhale, so we can really expand our thoracic cavity and allow for air to move into the lungs and that pressure to decrease. Um, So when we expire, we bring our diaphragm up, which is pushing our lungs up, right? We're pushing those membranes up. We're decreasing the pressure in the pleural space, which is this space right here, right? Pleural space. We're bringing our rib cage back in, therefore decreasing the volume, increasing the pressure, and we are able to expire air because we might even get a positive pressure. Um, right? So we might even get our pressure up to like +1 or +2, which is what we need to do to expire air. So we get our pressure in our lungs up to a positive number, and then we actually are able to force air out of our lung cavity. And that's how negative pressure ventilation works. And the way that it's made possible is all these membranes in our lungs working together to create these pressure gradients that pull and push and do all of these things. Something that's really important is our alveolus would, they have a tendency to want to collapse. So they're filled with water. They're very, very full of water everywhere. Um, and water has a really high surface tension, which we know because like bugs can land on water and sit there without falling through the water, right? So water has a high surface tension. So in order to fix that, we have to use something called surfactant. And surfactant is made in type 2 pneumocytes, which I have drawn in this diagram right here. So these are type 2 pneumocytes. And that is what makes surfactant. And surfactant goes in and actually it has a polar head. So this head is polar and this tail is nonpolar. We call it amphipathic because it has two opposing parts on it, right? And it sits and it binds to this hydrogen bond, and then it like sticks its lipid tail out into the alveolus, and it breaks up the hydrogen bonds. It makes them less tight. So it makes it so that um it makes it so that we don't have our alveoli collapse. Because if our alveoli all collapsed, then our lung would collapse, right? Because the air tubes are collapsing, and then we can't get air into our lungs, and we just would have big problems. And this helped us to explain I can't say this guy's name. Laplace's law? I'm gonna say it that way. Don't make fun of me if I'm saying it wrong. Which says that pressure in the alveolus is 2T over R. And T stands for tension, like surface tension from water, and R stands for the radius of the alveolus. Right. So the pressure in the alveolus is actually what is going to create that pressure gradient that allows us to inhale or exhale. Um, and so if we have a really big alveolus, it's going to be easier to inhale because the pressure will be lower. But it's harder to exhale because it's harder to get that air back out of our lungs because the pressure is lower, just because the alveolus is bigger, right? Um, if the tension is really, really low, then like if we have really, really low surfactant, the pressure will be higher because, wait, hold on. Yeah, the pressure will be higher because we, now I have to like really think about this. Actually, if the tension is low, meaning we have a lot of surfactant, the pressure will be lower. If the tension is high, meaning we don't have a lot of surfactant, the pressure will be higher, right? So these are all things where we could have something go wrong in the lung and you have to figure out like what's happening and why. Um, this surface tension idea is really important because without surface tension, our lung would collapse and we wouldn't be able to breathe. It would be really, really hard to um hold on. Now I have to think really hard. Without surface tension or without surfactant, it would just be hard to breathe. We would have a really hard time breathing. And for some reason my brain is not working as to why. Um okay. The alveolar gas equation is really important. Um it has really good uses clinically. So the alveolar gas equation teaches us that the pressure in the alveolus of oxygen equals, let me just write this down so that I don't lose my mind, and then I'll explain it. Okay, there we go. Now it's written down so I won't like have a hard time trying to talk and write. So this right here is the percent oxygen, the fraction of oxygen that you're inhaling. This is the pressure of atmosphere, so it depends on your altitude. This is the pressure of water. We have determined that the pressure of the water in your lungs is a constant. It's fairly consistent across people's. It's a, oh my gosh, my brain isn't working. It's a fairly consistent measurement across the board, right? So it's about point, oh, sorry, I'm doing the wrong one. It's about 50 millimeters of mercury. So we're going to subtract 50 millimeters of mercury from the pressure of the atmosphere, whatever that may be. PCO2, we know the normal PCO2 is 40 millimeters of mercury. So unless we're told otherwise, 40 millimeters of mercury is what we would assume in a normal, healthy person. And then R is actually a ratio. So R is a ratio of CO2 to O2. So basically it's a ratio of the amount of CO2 produced per O2 consumed. So what you're doing here is you're multiplying. Now I'm trying to remember if you multiply or subtract. You subtract this. Okay, well I killed that. Basically, what you're doing in this whole thing is you're subtracting the amount of oxygen we've burned, right? So that is going to tell us how much oxygen we've burned in order to tell us the pressure of oxygen in the alveolus. This is important because as we move clinically, right, the pressure in the alveolus is going to equal the pressure in the blood, roughly. It's not like exactly the same, but it's about the same as long as ventilation is occurring properly. So as long as we're moving air properly from our alveolus to our blood, then those two should be roughly the same. Um, if we aren't moving that properly, then we can start to think, okay, maybe we have an embolism, or we're experiencing an edema or something like that, where for some reason we aren't moving blood to our or air to our blood properly. Um, yeah, let's see what else. Okay, so how CO2 and O2 are transported in the blood. So this is the alveolus, and I'm gonna say that this blue is the partial pressure of oxygen in the alveolus, and this green is the partial pressure of carbon dioxide in the alveolus. And the partial pressures should be about the same in the blood. So if this is the venous side and this is the arterial side, the venous side, the PCO2 is about 45, and the P of O2 is about 40, I think. On the arterial side, the PCO2 is about 40, and the PO2 is about 98. In a normal human, the PO2 is 102 in here. And in here, it's about here, this PCO2 is about 45 as well. Now, oxygen and carbon dioxide are transported in the blood a few ways. Carbon dioxide can be transported. Um, I'm just going to draw, and I might have to erase and draw again for oxygen, or I can just tell you how oxygen is transported because it's really easy. It just happens one way. Um, Carbon dioxide is transported a few ways. One, it can be dissolved in the blood. So we can dissolve carbon dioxide in the blood. This is a very small percentage of our carbon dioxide. It's about 7% of our carbon dioxide is dissolved in the blood. So not very much of it is transported this way, um, but some of it is, right? So we do have a little bit of carbon dioxide in the blood. But a big way that we transport carbon dioxide is actually we attach carbon dioxide from the blood onto hemoglobin. So we attach carbon dioxide onto hemoglobin, and this is really important because it causes a shift in hemoglobin. So hemoglobin has two states. It can be in what we call the T state, right? So the T state, which is the tense state, or the R state, which is the relaxed state. And the tense state is the state where it does not bind oxygen. The R state is the state where it binds. And I'm not 100% positive that I'm right about that, honestly. I could be very wrong, which is a little bit concerning. Um, but I'm like pretty sure that that's right. Um, yeah, so we have the T state and we have the R state. And when we bind to hemoglobin for CO2, we change the state and we make it so that oxygen cannot bind. So we literally kick oxygen off of the hemoglobin. We tell it to leave. Um, and this is really kind of interesting because the affinity that oxygen has for hemoglobin can depend on the amount of carbon dioxide we have in our blood, which is important because as we pass by tissues that are metabolic and have picked up or have created carbon dioxide, and we pick up carbon dioxide in our blood from those tissues, we're going to dissociate oxygen faster and more effectively because carbon dioxide is going to bind to hemoglobin, and we're going to be able to release oxygen so that we can reoxygenate those tissues so that we don't become hypoxic, which is interesting. The other way that we transport CO2 is through this equation that we love so much. that we learned not very long ago. Um, right? So we have in our red blood cells carbonic anhydrase, a lot of carbonic anhydrase. And we create bicarb. So we transport bicarb in our blood all the time. But another interesting thing that happens when we create um, this equilibrium here, when we shift this way, when we have a great amount of CO2, is this hydrogen right here will bind to hemoglobin as well. And hydrogen also decreases hemoglobin's affinity for oxygen. So when hydrogen is bound to to hemoglobin, oxygen also unbinds. And CO2 and hydrogen don't bind till like the oxygen active sites on hemoglobin. They bind somewhere else, but they do bind and they increase um, the probability of the hemoglobin being in T state, and basically they kick off the oxygen. So the more that this stuff happens, the more that the CO2 is like here, the more that oxygen leaves. Um, on the converse, when we get to the alveolus, when we're trying to bring things into the alveolus, right? So when this stuff is moving into the alveolus, and we're trying to transport carbon dioxide into the alveolus, the carbon dioxide on hemoglobin is going to unbind. So this carbon dioxide goes away, it unbinds, and we're actually going to put it into the blood to replenish that 7% and keep it the same so that we still have this much in the blood. And we're going to put it into the alveolus as we expire when we take stuff out of the alveoli. The other thing that will happen is this equation, as CO2 decreases, will shift back this way, right? So we're going to have more carbon dioxide, which will also unbind these hydrogens and they go away, which increases hemoglobin's affinity for oxygen. So now our oxygens are going to come and bind to hemoglobin, which is kind of cool because it really helps us to know that there's more than just when the hemoglobin gets close to the lungs, all of a sudden we have this affinity for oxygen. It really has a lot to do with the other things that binds to hemoglobin when we're further away from the lungs, when the pH is lower, when we're more acidic, when we have more carbon dioxide present. All of those things add up to help the oxygen dissociate. The other thing that I guess I didn't really draw over here, I drew it at the alveoli, but the thing that I was going to draw here is that the oxygen levels in the cells are really low. It's like 20. So when your hemoglobin has oxygen bound here, what's gonna happen is they have PO2 that's pretty low, and then they have PCO2 that's fairly high. It's like 45. So that 45 concentration of PCO2 is basically going to come kick the oxygen off, just like I was talking about earlier with the PCO2 binding and leaving. I just, instead of putting it on two different diagrams like I was going to, I put it on one and made it kind of confusing probably. But that's like the idea with transport in the blood and how the gas exchange actually occurs. It mostly has to do with those interactions with each other and then their interaction with hemoglobin because a lot of the gas exchange or a lot of the transport is done by hemoglobin and then by the bicarb system for um, carbon dioxide. But the bicarb system also affects hemoglobin. Oh. Okay, so the last thing that we need to talk about is the hemoglobin dissociation curve. And this is the hemoglobin dissociation curve I have drawn. It's not perfect, but it's a good idea. Where basically what happens is when we are at 100% oxygen saturation, right up here, we're going to be fairly close to 100% hemoglobin saturation. And our bodies are really well adapted to make it so that as we drop in oxygen concentration or in our PCO2, right, we pretty good at keeping our hemoglobin saturated. So as we hit like 90, 80, 70, 60, 50, ignore the fact that there's an extra line, right? We we keep our oxygen pretty saturated, but at that like 40 mark, our slope really makes a curve change, and we really start to desaturate that oxygen or that hemoglobin with oxygen. And we start to really be able to pull oxygen off really, really fast. And some things that can change the shape of this dissociation curve. So like we can make a curve that looked more like this, or we could make a curve that looked more like, okay, that one was like a really bad drawing. Let me try that again. It won't look like that. It'll follow the shape of the curve still. It'll just be like a little bit less. Or that looks more like that. Right? So if we want to increase our ability to saturate our um, hemoglobin, we could decrease carbon dioxide because carbon dioxide is what kicks the oxygen off. So if we have less carbon dioxide, oxygen will stay on hemoglobin for longer. We could increase pH, which is also decreasing hydrogen, right? Because our oxygen is also kicked off by hydrogen binding. So when hydrogen binds, we kick oxygen off. We also could decrease temperature. We didn't really talk about temperature, but temperature has an effect on gas exchange. And when we increase temperature, we do release oxygen faster. So, like if you have a fever, that's why when you have a fever, you get winded really fast. Like you stand up and you're so tired because your oxygen is being pulled off your hemoglobin a lot faster than it would be if you were at normal body temperature. But if you were to decrease your body temperature, then you would stay saturated longer. Another way that we can do this is with fetal hemoglobin. And the reason that this works is a really cool molecule. It's called 2,3-BPG. And 2,3-BPG has this really cool affinity to bind to hemoglobin. And it binds to hemoglobin and it kicks oxygen off way faster. So it binds to all four chains rather than just one. Like the carbon dioxide can only kick one oxygen off. 2,3-BPG binds to all four chains and kicks oxygen off hemoglobin. Now, fetal hemoglobin, they lack the ability to bind BPG because they have a different, or 2,3-BPG, sorry, because they have a different um, chain. We have beta chains. Fetal hemoglobin has something called a gamma chain instead. It's just a little bit of a different chain. So there's two amino acids that bind to 3-BPG, and fetal hemoglobin lacks that. So they, the fetus, actually can steal oxygen from its mom. It's kind of ingenious the way that that was designed, but they have the ability to hold on to oxygen a little bit longer than somebody with adult hemoglobin. Um, and even babies have adult hemoglobin. Just we have fetal hemoglobin when we are in the womb, obviously. Things that would create the yellow line are like the opposite of the things that I have written above. So if we increase our CO2, right, we're going to bind CO2 earlier, oxygen gets kicked off earlier. If we decrease pH, which is increasing our hydrogen concentration, that'll do it for us. If we increase our temperature, that'll do it for us. Any of those things. Another thing that we can do to kick oxygen out faster actually is exercise. But again, that's probably just the whole increasing your body temperature thing. Maybe it's increasing blood flow. I'm not sure, but I do know you can exercise and that will decrease your oxygen hemoglobin dissociation curve. Um, yeah. Oh, also the other thing that I forgot to mention is that the only way that we transport oxygen in the blood is through hemoglobin because oxygen has a really, really low solubility. So it's really hard to dissolve oxygen in the blood and just like carry it like we can carbon dioxide. So we pretty much only, actually, we exclusively carry it on hemoglobin. That's pretty much it. Whereas carbon dioxide, obviously, we can carry it a few different ways. But with oxygen, we really just carry it on hemoglobin. Um, which is why it's so important that we're able to kick it off of hemoglobin. Because if we can't, then we can't oxygenate our tissues, and then we have a big problem because we start getting hypoxic in places that we don't want to be hypoxic, which would be really bad. Um, yeah, that's kind of the idea of the oxygen hemoglobin dissociation curve, some factors that affect it, and which way it affects it, and